So thank you all. Thanks for the organizers for giving me the opportunity to speak here today. So my name is Gilad. I'm from the Baker Institute at Cornell University at the lab of Charles Danko. And I'm very interested at the interplay between genome architecture and transcriptional regulation. So today my story is about enhancer promoter contacts in the genome, and how they are facilitated by factors in the transcriptional machinery, such as RNA polymerase II itself and PARP1. So a little bit about metasomal transcription. The early steps in metasomal transcription involve the recruitment of POL2, which is the responsible for transcription of most genes in our genomes, to the transcription start site of enhancers and promoter, which is phosphorylated on its C-terminal domain, which is highly, intrinsically disordered and plays an important role in its regulation. And then it starts transcribing. About 20 to 60 base first downstream to the transcription start site, POL2 pauses for reasons that in terms of function, we still fail to understand, but we do understand that it's highly conserved across metazoan. And we know that it's regulated by these two factors, NELF and DSIF, as well as another factor, PTFP, that is responsible for the release of POL2 into productive elongation by phosphorylating DSIF and NELF, as well as an additional residue on the C-terminal domain of POL2, removing NELF from the complex, and POL2 sends POL2 into productive elongation. And we can follow these thread of events using a nascent RNA sequencing techniques like PRO-seq, where we can follow the density of transcriptionally engaged POL2 across the genome. So we can spot transcription start sites of gene. We can spot the density of POL2 at the pause position, and also the density of productively elongating POL2 at the gene bar. But there's another important layer of transcriptional regulation in metazoan that is really important for organismal complexity in metazoans, and that is cis transcriptional regulation. It's facilitated by chromosome folding, and particularly by contacts between enhancers and promoters that can reside kilobases to megabases away in terms of genomic distance, but come close together in 3D space. And this proximity between enhancers and promoters, we and others have shown that is critical for the ability of enhancers to activate transcription from target promoters. However, since both of these elements in the genome are transcribed, we've hypothesized that perhaps these early steps in transcriptional regulation can also shape enhancer promoter contacts. So to ask that, we've collected a lot of microarray data that gives us contacts across the genome in nucleosome resolution. And we do that by digesting the chromatin of fixed cells with microparticle nuclease to get the nucleosome resolution. And then we use proximity ligation to capture everything that is close proximity. And by capturing these ligation junctions, we can map those reads into to the genome, where each side of the read will map to different locus in the genome. They could be, again, kilobases to megabases apart, but close enough to ligate in the genome. So we can present these contacts in these contact heat maps, where the x-axis here is the genome sequence, and the intensity of the color represents contact frequency. And you can see, for example, this dot here represents high contact frequency between this locus here and this locus here. So here we compared between two different cell lines in human, representing lineages of human hematopoiesis, and we compared both contacts and nascent transcription, and you can see how these things correlate in the genome. So if you look at this region, or this topological associated domain, you can see that it's more transcriptionally active in your case, and you can also see that it is reflected with more contacts between regulatory elements. You can see just the mirror image if we take just one topological associated domain to the left, where K562 show higher transcription and higher contacts. So overall, just by eyeballing the data, we can clearly see that transcription and contacts are correlated. However, can transcription actively facilitate enhancer promoter contacts? So to answer that, we've looked at mouse embryonic stem cells where transcription was acutely inhibited either by triptolide that inhibits the recruitment of POL2 into enhancers and promoters, or by flavopyridol that inhibits the release of POL2 from the pause position into productive elongation. And by looking at microarray data and normalizing for background contacts, we can see that most enhancer promoter pairs in the cells, in the genome, lose contacts between them, as represented here in dots in the scatter plots, at the treated samples compared to the DMSO control. We can also show that this effect is specific to enhancer promoter contacts. When we look at other major anchors for contacts in the genome, like CDCF binding sites, we did not see a similar effect, indicating that transcription is not only the substrate of cis regulation, but also actively shape enhancer promoter contacts. So what is it about transcription, and specifically about this early step in transcription regulation that can do that? We first looked at initiation, and we can appreciate the level of initiation by looking at the level of POL2 at the gene body. And following initiation, different initiation rates with different promoters, we've noticed that initiation overall is correlated with an increased contact frequency between enhancers and promoters. But this is not specific to any region around enhancers and promoters, and we can see that the whole aggregated contact map seems to light up. However, when we specifically look at the changes in the post-POL2 density, then we saw that the increase in contacts is specific to the points of contact, or the focal contacts between transcription start sites of enhancers and promoters, you can see here. That indicated that perhaps pausing stabilizes enhancers upon proximity rather than increases their movement towards each other and the frequency of contact. So to get another look at that, we also compared between the different cell lines, as I've shown you before. And after controlling for the level of initiation, we saw that an increase in pausing is a correlate between the different cell lines, also correlated with an increase in enhancer promoter contacts. So we hypothesized that POL2 pausing plays an important role in stabilizing enhancer promoter contacts. To test for that, we've depleted one of the subunits of NELF, which is important for the proper pausing of POL2, by a rapid and reversible Degran system. And we saw that after as little as 30 minutes, we can spot the depletion of post-POL2 near transcription start sites of both enhancers and promoters. So we generated this time course where we've depleted NELF-B and post-POL2 and then regained them for 8 hours and 24 hours. And we got exactly what we expected. As post-2 was depleted, post-POL2 was depleted, so were enhancer promoter contacts. And then as post-POL2 was regained, so were enhancer promoter contacts. So we can conclude that post-POL2 is important for the stability of enhancer promoter contacts. So to get into the molecular mechanism, we searched in the literature, and we saw that based on a lot of recent microscopy studies, a recently suggested model claims that the contacts between enhancers and promoters are actually stabilized through interactions between factors in the transcriptional machinery in high local concentration through intrinsically disordered regions, such as ones that we see a lot in transcription factors, but also global regulator BRD4 and mediator, and including POL2 with its C-terminal domain that is so important for its regulation. So based on this model, we suggested an additional important factor that might explain the connection between POL2 pausing and enhancer promoter contacts, and that's poly-ADP ribose polymerase 1, or PARP1, which is responsible for about 90% of PAR chains in the cells. It was shown to bind almost all active enhancer promoters in the genome and parallate NELF, which enhances its release from the post-POL2 and post-POL2 release into productive elongation. So considering the effect that I've shown you of post-POL2, that effect of PAR1 should destabilize enhancer promoter contacts. However, PAR chains themselves were shown to enhance contacts between different proteins through enhancing contacts between intrinsically disordered regions. And based on the clustering model, that effect of PAR1 should enhance enhancer promoter contacts. So we've inhibited PAR1 for 90 minutes using olaparib, and we saw exactly that. So we saw a global decrease in enhancer promoter contacts, which is in line with this clustering model and the contribution of PAR1 and PAR chains to the stability of this transcription-associated protein. However, we did also see the accumulation of post-POL2 following PAR1 inhibition, which is in line with the literature, and we wonder whether this effect of PAR1 inhibition will antagonize the effect that I've shown you earlier. So we've correlated this effect with the effect on enhancer promoter contacts, and what we've seen is that as enhancers and promoters gain more post-POL2 signal at the pulse site, they lose significantly less a smaller amount of contacts between them, indicating that, in fact, PARP1 can also destabilize enhanced promoter contacts through its action on post-POL2. So to summarize everything that I've shown you that post-POL2, which is so far was not very understood in terms of function, but it's very common across metazoan, seems to stabilize enhanced promoter contacts. I've also shown you that PARP1, which is an important chromatin and transcription regulator, seems to stabilize enhanced promoter contacts globally as well. However, through its action on post-POL2 and the release of post-POL2 into productive elongation, it seemed to also have a destabilizing effect in steady state. And with that, I would like to end and thank my lab members and everyone that contributed to this great project and the funding agencies. And thank you all for listening. I'll just say that today was just the tip of the iceberg. So if you're interested in the full story, please go to the bio archive. Thank you.